Thank you very much for this honor to be here and presenting our talk on experimental pathology regarding some aspects of Alzheimer's disease. As rightly pointed out by Nathan, there are various theories and people stuck with them. But being an experimental pathologist, I have the freedom and liberty to find out the balance, and we do not belong to any particular theory but looking some facts on the basis of experiments that we observe. Before I go, I can tell you that this is a multi-organizational collaboration working since 2003, and we have great opportunity to work along. And I should tell first that whatever I am speaking today, it is not my official version or conclusion. All conclusions taken or presented is my personal opinion. We are working on nanomedicine when we came in contact with US Air Force Research Laboratories in 2003, trying to find out how disease can be modified by the presence of several tiny nanoparticles that may be still present in this house. The second point is that no disease comes alone. Every disease, whether it is stroke, dementia, Alzheimer, you are the clinicians, you know much better. It is always a combination of disease in human beings, and therefore each factor can influence the other factor. So that's why what we need not a single drug, but a multi-modal drugs, and our experiment is still going on. I started my work on blood-brain barrier because I still believe that blood-brain barrier is unique to the brain. It protects for all kinds of adverse effects. But on the other hand, for neuropharmacologists, could be disease states, this blood-brain barrier could be a hindrance. So we have to also hear a balance. And I greatly owe to Professor Stanley Irwin Radford, because who published in 1976 the first book on blood-brain barrier in physiology and medicine. And I was so impressed that I did a thesis in Banaras Hindu University in India, understanding how stress can modify the blood-brain barrier, and my supervisor was Professor P.K. Day at that time. So here, I am more fascinated about the endothelial cells of the cerebral blood vessels. They constitute the blood-brain barrier. Of course, we have neurons. We have to protect neurons because neuroprotection, the term was coined in early 80s. And then people believe that we have to protect neuron no matter what else is there. But now we know that glial cells are even much more important, and so as endothelial cells. We try to highlight this in one of our earlier books, and I was privileged that Milton Brightman from NIH wrote the foreword. I tell you why. This is the first example of the tight junction as an anatomical site of the blood-brain barrier by lanthanum, and this is discovered by Brightman and Reed at NIH. As I told you, I am basically a neuropathologist. My mentor is Professor Ingrid Olsen, and later on I understand electron microscopy with Dr. from Germany. He was one of the leading stars in neuropathology from Europe. I still believe in the theory that all kinds of insults to the blood-brain barrier leads to different kinds of problems. Now coming to nanomedicine and Alzheimer's disease, I am so happy that Dr. Heiss has explained everything to you, so I will cut short this part anyway. This is very well known that it was discovered in a lady, and from pathological point of view, the Alzheimer brain looks like this. The temporal cortex and other areas are just shrunk, and therefore it is quite understandable that those functions commanded by those areas were also disturbed or abolishhed. The pathological picture of senile bliss you have already seen, so no need to discussion, and also this amyloid beta peptide. The problem is that we must understand that these amyloid beta peptides are also present in the plasma. And then, depending on the situations, they can enter into the brain. There is another professor who is very leading in Alzheimer's disease, Verislav Zokovich, and he feels that the expulsion of amyloid peptide protein from the brain is also important. So these are two different theories. But why I concerned the opening of the barrier in these diseases that was much less focused earlier. This is again telling I do not believe to either amyloid theory or tau theory, but what we are looking is a collective way. Because in nature you have a balance and disbalance. So you can see, as told by Dr. Hayes, that tau is very important, and there are various ways they are released. This cartoon I am showing only that not only neurons, endothelial cells are also equally important in disease as well as the glial cells. So I searched some literature regarding blood-brain barrier disruption in Alzheimer's disease, and I am showing some of the examples here. You can see that indeed the blood-brain barrier is altered in Alzheimer's disease. Here are some cases of 300 cases, and they were presented by Dr. Bavaria, and you can see that extravasation of serum proteins can be seen very frequently in Alzheimer's disease. You know that in brain we have almost everything that is found in the blood, but there is no protein, albumin, and also in CSF. So whenever the CSF albumin or proteins are high, we have disease, either psychiatric or any kind of disease. So you can see that serum protein was found, means blood-brain barrier was altered. There are also other enzymes that are responsible for blood-brain barrier. They are found in the endothelial cells, and they are also altered in Alzheimer's disease. So the point is that blood-brain barrier is an important part also in Alzheimer's disease. Another example, pathological changes in endothelial cells. They were described since 1993, and it is quite good, but still ignored. Then we came about the treatment. There are various ways you can treat to take out one factor or the other. But the point is that if we use clinical cases, can it benefit? Probably not, because the clinical cases are so complex, depending on so many other factors together. So therefore, we decided just to understand some experimental model. Then also the neurotrophic factors came into the play regarding the Alzheimer's disease treatment, and I summarized some of the words here. Then the point is the nanotechnology. Nanotechnology came quite recently, and the idea is that any drug, if it is delivered through nanotechnology, can reach to the different parts of the brain quickly and may stay there. It could be like slow release of pumps inside the brain, and the drugs cannot be destroyed because they are bound to certain aspects of nanoparticles. But the problem is still there. In every aspect we have problems. So we decided just an example to use Cerebrolysin because it is a multimodal drug and a mixture of different neurotrophic factors, and tested our hypothesis in some other model like traumatic brain injury. And here we show that in an animal model of traumatic Alzheimer's disease, we can see that this leakage of serum protein was reduced with Cerebrolysin treatment. So this would be one of the earliest indicator in our hand that Cerebrolysin given is able to reduce the blood-brain barrier leakage to serum protein in a model of Alzheimer's disease. Then there are various ways of nanotechnology to induce drug. We have chosen nanowire delivery because this was earlier discovered by Dr. Young. He was an electronic person. And we are using in biology and medicine nanowires. Titanium at the moment we are using. This looks like this. And this is Dr. Ryan Jian and Dr. Wang. They have the US patent on nanowire technology to label drugs. So we are using in a collaboration of these two people. Nowadays, we also heard about lots of stem cells, and people are thinking that stem cells would be cure for almost every disease, but these things are not come true. But I can tell you that even stem cells are nowadays nanowire, and they can live longer and protect the brain anyway. We are working in Arkansas because this is a multi-million-dollar project since long time, and at that time the previous Democratic governor was very supportive to us. We still get support from the government in various aspects from the United States and of course the state of Arkansas. And everybody can ask question, what is nanowire delivery, and are we fooled to use this nanotechnology and trying to fill the data by publishing some rubbish? So everybody wants to test their own hypothesis very crucial way. And you might be knowing that every year in the United States, this National Innovation Summit occurs where you can submit your innovation. So we challenged our own hypothesis by ourselves and submitted one of the innovations. My wife Sharma has submitted this, nano delivery of cerebrolysin and combination with medium time stem cells. We know that traumatic brain injury or even other cases, they are much more different. So we are surprised. The Committee of National Innovation found it, this is one of the top 15% technological innovation to this drug delivery. And you can see that we are very happy that at least we are going in the right direction, no matter whatever our critics will say. And regarding the nano delivery of cerebrolysin, we tested in 2013 against neuropathic pain in animal model. And at that time, they found that it was top 20% technological advances. So, what we do, we examine amyloid beta peptide reactivity in animals, and you can see that this is increased. And when we have cerebrolysin treatment in this model, it is much more better if we deliver nanovirus cerebrolysin. I'm not going into the theory of amyloid is really cause or effect of the Alzheimer's disease. Then we tested in another way. We measured amyloid beta, and we can see that nanovirus cerebrolysin here, it is much more effective in reducing these changes. The second thing happened also with tau. When we have delivered nanovirus cerebrolysin, it is much more effective than the cerebrolysin alone. So we continue to use this model of intracerebral ventricular administration of amyloid beta peptide. This is a well-established model, and I'm sure that it cannot produce the Alzheimer's disease in clinical condition, but it can have some relevance to the pathological aspect. Because you cannot produce any experimental model with pathological cases. Even traumatic brain injury, when you do animals, you have to anesthetize them, and in humans, you get traumatic brain injury when you are conscious. So you cannot do that. So look these cellular structures. What we can see that this amyloid infusion, this amyloid beta peptide is there. Nanovirus cerebrolysin has significantly reduced as compared to cerebrolysin. And here is the albumin deposition. So this suggests that blood-brain barrier is leaked here, and even this nanovirus cerebrolysin is doing much better as compared to cerebrolysin alone. Then we also studied this astrocytes, I told you, and this is only untreated group, and nanovirus cerebrolysin, the astrocytic reaction was also reduced, and here is the neuronal changes. Then we went to electron microscopy because we believe that we should see the cells. And at this time, we focused on myelin vesicillation. This is one of the important aspects of neuronal damage. And here you can see here that nanovirus cerebrolysin is doing better. Even cerebrolysin was good, but nanovirus cerebrolysin delivery has better effects in our hands. We know that Alzheimer's disease is a multifactorial effect, so we also try to mix those things. Animal model of diabetes and traumatic injury. And as you can see here, that in situations of diabetic cases, the titanium dioxide nanovirus cerebrolysin is doing much better, either insulin, TFFP, or even albumin. Head injury is also, in last 5 minutes, I can tell you that our soldiers who get head injury, it is already known that eventually they will develop Alzheimer's disease. So what are the factors doing that can promote Alzheimer's disease in head injured patients? Even the physical effect of head injury is not seen. We applied this model of concussive head injury because that is quite common, of course, in that model. And you can see that this model appears to be working because whenever this concussive head injury occurs, many clinicians can know that it's counter to phenomena. Injury occurs is much more greater in degree on the other side. And this is also true. We have injury on right side, and we can see much more injury on the left side. I just showed you some of the example here in the table that only tell you that nano delivery of, here we have also combined with the stem cells with cerebrolysin, and here is amyloid beta peptide infusion, traumatic brain injury, and you can see that the values, even deposition of the beta peptide and albumin cells were much less as compared to both the elements given or the drugs given alone. So the combination of different kinds of things, they can potentiate each other's effect. That is the summary of this geometry table. And I can show you here the example, pathological example. Nanoviral delivery of cerebrolysin plus mesenchymal stem cells has the best effect here. Last 3-4 minutes I will talk about neprilysin, because I told you that each and every factor can contribute to each and every disease. And whatever we see in a pathological situation, its expression, we still do not know that these are the cause or effect. But whenever we see by the drug effect something is going down and we are getting better, we can understand that this could be one part of the large image. Neprilysin is now recently known that it also increased and decreased both in brain injury. There are some theories that neprilysin is protective. Even there are some theories that even upregulation of amyloid can also be protective because it is doing something good. So, and here is an example that neprilysin is very close to the amyloid beta peptide but does not match with GFA. So what we did? We nano-labeled neprilysin and nano-labeled cerebrolysin and injected together and compared. You can see here that this is the nano-wired cerebrolysin plus amyloid beta peptide infusion, and this is the combination. What you can see that the leakage of blood-brain barrier is also decreased after traumatic brain injury combination. Sorry, just to show you. And here, every parameter is reduced by the combination of nanoviral cerebrolysin and amyloid beta peptide. This is an example of hippocampus, because hippocampus is highly vulnerable, as you know here, and this is traumatic brain injury plus our model has much more damage, and when we have given cerebrolysin and neprilysin, the cells were highly reduced. Means the damage is reduced and we have neuroprotection. We are working now with Dr. Proto. He has some idea how we can enhance the effect of nanoviral cerebrolysin with some of the ubiquitin inhibiting enzymes, and also some other events we are working on that. So I can only say that nano delivery of drugs potentiate the basic effect of the drug, and even we can use other factors to combine which are neuroprotective. They can have additive effects. In last 3 minutes, I can only tell you that people are talking about, can we deliver the same drug with 2 different kinds of nano delivery, and can we get the same results? Here we tested PLGO level cerebrolysin and titanium oxide nanoviral cerebrolysin. What you can see here, that although both were neuroprotective, but the titanium nanoviral level cerebrolysin was much more effective in reducing blood-brain barrier in different areas in Alzheimer disease model. So there is something that we also understand the delivery. Of course, we are also working with Dr. Shelley on combination of CRF fragments together with cerebrolysin. Can we enhance the effect of each other? But these are still undergoing on. This is Dr. Asia. She is labeling another form. We have now labeled cerebrolysin nanospheres and deliver. And this model is quite, you can see, can release cerebrolysin steadily over the period. And now also even the question is asked by Yonos that how we can enhance the antibacterial drugs in the case of this resistance. Can we also use nanovirus? And hopefully we will have some results in this aspect just for our neuropharmacological questions. So in conclusion, we can say that nanoviral cerebrolysin is quite effective, and its effectiveness is much more increased when we combine with some other drugs. Of course, there could be some other drugs they can decrease, but so far we are not studying. I am very happy that Society for Neuroscience has accepted Dr. Murasam's policy paper when people are talking that we must understand that nano delivery of drugs and different kinds of biological effects should be needed to explore. And this is another sign that maybe we are right. This PLG nanoparticles loaded cerebrolysin and Alzheimer's disease is a symposium, a nano symposium, Society for Neuroscience. And even this genetic nanosphere, I showed the picture of Dr. Asia. Her work against Parkinson's disease is also accepted in nanosymposium, so probably we are doing right. And there are also some other papers, I'm not going in detail. So I believe that in our hands, nanomedicine could be the future, and we have to see its nanoneuroplasticity. That is very important before it comes to clinical practice. There are lots of supporters in this organization, and many organizations support this work for a long time. My collaborators, and this is a presentation from Uppsala University, 1477. It was established. Some say that before the advent of the United States of America. Thank you very much for your time.
Thank you. Are there any comments or questions from the audience? Okay, that means you continue to work. Can you give us some insight as to why you believe, why the nano delivery of Cerebrolysin, this other agent, provides this additive benefit? Are you increasing biological half-life? Are you delivering it over time? What is the mechanisms that underlie this?
Yeah, that's a very good question and quite expected. We have just published a review in CNS and drug disorders a few days ago, and based on this, if you have this nanowire delivery of drugs, the drug is bound around the scaffolds of the nanowires, and there are evidences that these nanowires pierce the endothelial cell membrane or any cell membrane in vivo, and they can enter without damaging them, and they can then either the intracellular space or interstitial space, and then they release these drugs. And of course, the nanowires can stay there. That is another part. Up to 6 months in our hand, these nanowires itself do not do any pathological changes. But the point is that the burning question is that what is the fate of these nanowires, and then how long they can be innocuous. But in the University of Arkansas, they also do this deposition in the knee surgery, these nanowires covered with drugs. And they have 6 years. The patients do not complain anything, and the recovery is much faster. That's the only thing I can say at this moment.
Is there any difference between the wires and the globules?
The wires and?
Oh, these globules.
Yeah, we tested this together with our friend in Italy. So we use this soft nanoparticles inside level Cerebrolysin. It is also quite effective than the drug alone. But in our tested model, the magnitude of effect was still better than titanium nanowire delivered Cerebrolysin. We still do not know the difference.
Yes, please.
Thank you very much for your presentation. Do you have any other example of delivering neuroprotective drug by using nanotechnology? Successful examples?
The question is, do I have...
Other than Cerebrolysin?
Yes. This is a very large project, and we are testing, our object is nano delivery of drugs. From our published work, we have also delivered nanowire ondansetron in some aspects of this is 5-HT receptor antagonist in some aspects of drugs of abuse. We have also delivered H2-9051, this is an Astra drug antioxidant in other forms of traumatic brain injury, and then there are also some other drugs still under investigation. My point is that any drug that is delivered using nano delivery technique that we are using has much better effect than the drug alone. And you can find this nano delivery of drugs in the literature. There are a couple of items, 70 papers available now.
Okay, so I thank you very much again.
Thank you, sir.
And we carry on. Our next speaker is Dr. Alvarez from Spain, where the speaker is working in the field of neuropsychiatric disorders, diagnosis and treatment, and today is going to speak about BDNF modulation for the treatment of neurocognitive deficits and the prevention of dementia after TBI.
Thank you very much, Roger, for your introduction. And distinguished